Assalamualaikum and good day, everyone. Now we will begin our new unit, which is unit number 3, prevention of infectious diseases. So we will start with the topic number 1, which is immunization. This is a very short topic, okay? For this topic, the specific learning outcome is to describe the vaccination strategies which intended to reduce the risk of vaccine-preventable diseases in the oral healthcare setting and explain the immunization program in Malaysia. Okay, for the definition, for immunization is the process whereby a person is made immune or resistant to an infectious diseases, typically by the administration of a vaccine. And the second one, vaccines stimulate the body's own immune system to protect the person against subsequent infection or disease. And the third one, a vaccine is antigenic but not pathogenic, means that it's anti the antigen, but it's not going to cause any disease. Okay, that's what a disease vaccine means. All right,
Vaccines
let's
and the immune
watch
response.
this video.
How vaccines work. Influenza vaccines are able to trigger an immune response by mimicking viral infection. They are usually manufactured using inactivated or killed virus particles taken from various circulating influenza strains. Inactive fractioned viral components contain the subvirion particles hemagglutinin, HA, and neuraminidase, NA. In Canada, influenza vaccines are administered via intramuscular injection. These fractioned particles containing the foreign antigens hemagglutinin and neuraminidase are released from the vaccine into the bloodstream. There, they are met with an immune response mediated by various immune cells, including macrophages, T lymphocytes or T cells, and B lymphocytes or B cells. A major mechanism of action involves macrophage phagocytosis of hemagglutinin, breaking it into smaller components. After ingestion, macrophages display the hemagglutinin antigen on their surface in combination with a specific receptor known as the major histocompatibility complex, or MHC. T cells are now able to recognize and bind foreign antigens that are associated with the MHC. Upon binding to the MHC receptor, T cells become activated and proliferate into either cytotoxic T cells, regulatory suppressor T cells, or helper T cells. Activated helper T cells express hemagglutinin receptors specific to the vaccine strand on their surface and play a major role in antibody generation and memory B cell activation. Unlike T cells, B cells are able to ingest hemagglutinin independent of the MHC. Once internalized, B cells process the hemagglutinin antigen and present it on their surface in combination with an MHC. When activated helper T cells interact with activated B cells expressing antigen MHC receptors, they begin secreting lymphokines, which have several effects. Lymphokines trigger activated B cell proliferation, which leads to either their differentiation into memory B cells or into plasma cells. Plasma cells produce hemagglutinin antibodies specific to the strain of influenza contained in the vaccine. Memory B cells aid in future immune response when exposed to an active influenza virus. When an infected host sneezes towards an uninfected person, the nasopharynx is exposed to aerosol droplets containing whole, live influenza virus. Once inhaled, the influenza virus attempts viral colonization of nasopharyngeal epithelial cells. Assuming that the vaccine strain matches that of the live virus, hemagglutinin antibodies block viral attachment of live influenza virus to host epithelial cells, and overall disease is either avoided or diminished in severity.
Okay, so next one, looking at the immunity itself. So as you probably have gone through this topic, for the specific defenses immunity, we have active immunity and we have passive immunity. So for passive immunity, it can happen natural or acquired, similar to active immunity. The transfer of maternal antibodies through placenta and also the transfer of maternal antibodies through the milk would be a natural part of the passive immunity. For the acquired immunity, it has the after the administration of immunoglobulin or anti-serum. Okay, for the active immunity, the natural one would be the clinical infection after the clinical infection or even subclinical infection. All right, for the acquired one, we give vaccines. All right, so that is the active immunity. So for the vaccination program, we actually using the active immunity category, which is the vaccination. Okay. Right, so this is just to summarize what the previous slide talking about. So looking at the immunity itself, so you can probably read this later. Just a little bit of a diagram. All right, so let's look at the immunizing agents. The first one is vaccines. That the one is quite popular everywhere. It is made from the very small amounts of weak or dead germs or viral or bacteria that can cause diseases. Also toxins sometimes. Okay, but there is another one, immunoglobulins. The two types of immunoglobulins preparation are available for the passive immunization, which is coming from the normal human immunoglobulin or the specific human immunoglobulin. Okay, it's called hyperimmune. The third one is anti-sera, which is the materials prepared in animals or non-human sources such as horses. Okay, so these are the three types of agents. You probably know or heard of vaccine before, but there are other two options, which is the immunoglobulins and anti-sera. So make aware that there are three agents available for immunization. Okay, the most common one is still vaccine. Okay, let's discuss or look at the types of vaccines. The first one is polysaccharide and polypeptide, which is the cellular fraction. Number two is inactivated or killed vaccines. Number three, attenuated live vaccines. Number four, live vaccines. Number five, toxoids. And number six, surface antigen, or what we call recombinant vaccines. Okay, so there are six types of vaccines that are available for us to use. All right, when we give the vaccines to our patients, the routes of administration is different for every type of vaccine. For example, if you look at the common one that everyone is used to, which is the intramuscular vaccination, IM, you can see hepatitis B, IPV, PCV, tetanus, diphtheria. All these packages are given through IM. There are other things using other vaccines using different routes, and everyone had a BCG done, which is given through intradermal, measles through subcutaneous, as well as yellow fever. And there are oral vaccination as well, but not very common for us, which is rotavirus and OPV. So these are the available routes of administration for vaccines. Okay. All right. This is the last slides. One of the last slides. Okay, this is the immunization schedule in Malaysia. This is taken from the Ministry of Health website. If you look at the diagram here, so it starts at birth, so all the way to age of 15, where BCG is given for the first dose at birth, and then all the way to 15 years old, which we will give a tetanus booster. So you can see the whole range of vaccines been given to an individual since birth all the way to age of 15. But beyond age 15, there are other vaccines that can be given. For example, flu virus. So there are uh, flu virus and meningococcal vaccination also available, okay, for adults. For example, if you want to visit other countries, sometime that is required for you to have certain vaccination be prior to the travel. Okay, so this is for the Malaysia situation. Okay, so if you go around the world, they would have almost similar schedule, immunization schedule, and this schedule also proposed, similar proposed by the WHO. Okay, so that's it about immunization topic. Hopefully you understand what trying to say here. All right, thank you.